Cells are the basic unit of life. As such, they're the structural unit of all living things. There are three major components to cells. We have the plasma or cell membrane, which is the boundary of the cell, the cytoplasm, which is the site of all the chemical reactions that occur in the cell, and the nucleus, which is the control center of the cell. This is a generalized cell, meaning it has some of everything. We can see the cell membrane. Here in the cytoplasm, there's all kinds of interesting things. And then here is the nucleus, and the nucleus has some subcomponents as well. In addition to the cell, there is extracellular material. One of the things are the body fluids. This is collectively referred to as the interstitial fluid. This is going to bathe the cells in material. Because our body fluids are primarily water, and water dissolves almost everything, body fluids are a way to transport dissolved materials over the cells. Blood plasma and cerebrospinal fluid are a couple of examples of body fluids. Cells also secrete various things. Enzymes, which are cellular catalysts, may be secreted into the body fluids. And mucus is manufactured by some cells to help lubricate the surface of the cell. And finally, we have the extracellular matrix. This is a collection of proteins and polysaccharides. These can mesh together and help bind cells together. The plasma membrane is referred to as a fluid mosaic. It has primarily a phospholipid bilayer. Now you remember that phospholipids had that polar head and the nonpolar tails. And when you put phospholipids in a watery solution, they line up heads out, tails in a double layer. About 5% of the cell membranes are glycolipids. These are on the outside of the cell membrane. Glycolipids are literally sugar fats. About 20% of our cell membrane is cholesterol. This is interwoven in the lipid tails and helps stabilize the membrane. And then we have the protein components of the cell membrane. The integral proteins are transmembrane proteins. That is, they go all the way through the membrane. These make things like channels, receptors, enzymes, and carriers. Then we have peripheral proteins. These are loosely attached to the surface of the cell. Many of these are enzymes. Many enzymes need to be attached to a membrane in order to function, and so enzymes may temporarily attach and then detach when they no longer need to be active. These peripheral proteins can also be involved in cell shape changes and in mitosis. And then finally we have a layer of glycoproteins. Again, these are sugar proteins. Here we see the depiction of the cell membrane. Here are those phospholipid, that bilayer, heads out, tails in, and this makes up the majority of the membrane. We have the integral proteins, the ones that go all the way through, and we have some peripheral proteins, some that are just attached to the surface. Here are the cholesterol, helping to strengthen the membrane. And then these are the various glycolipids and glycoproteins that help make up the sticky surface of the cell. The cell membrane is arranged so that the extracellular fluid, the watery environment outside the cell, cannot readily mix with the watery environment inside the cell. The membrane proteins have several functions. Some of them are transporting molecules. They either act as channels or they are involved in active transport. Some of them are enzymes. They allow molecules to interact and either come together or split apart. Because these are proteins and proteins all have a specific shape, they may act as receptors. That is, they may have a specific molecule that fits in them, and once this receptor is activated, some sort of message can be transmitted to the inside of the cell. This signal molecule never has to enter the cell. Again, because proteins have specific shapes, they may fit together sort of like puzzle pieces to help hold cells together. Those integral proteins are in the cell membrane very tightly, and so structures on the outside or on the inside of the cell can bind and have some stability. And then finally, again, because of that specific shape, we may have cell-to-cell recognition. Two cells may be able to interact because they recognize each other at this protein junction. The glycocalyx is the sticky cell surface. This is a function of the glycolipids and glycoproteins that are secreted by the cell. Each cell will have its own unique sugar arrangement. This helps with cellular identification. These are sometimes referred to as biological markers. Each individual person has their own biological markers, and each individual cell has its unique biological markers. The sperm is able to find the egg. Even though it swims past thousands of other cells, it will only attach to the egg. That's because of the glycocalyx. Also, the glycocalyx is how immune cells identify foreign cells like bacteria, or can even identify damaged cells. Damaged cells will change their glycocalyx and send out a different signal. Cancer cells will alter their glycocalyx. Cancer cells can be identified initially by the immune system and destroyed. If the immune system can't act rapidly enough, cancer cells may grow to large numbers and we have tumors. This is an example of a glycoprotein being recognized by the protein on another cell. Cell junctions are membrane junctions that secure cells and hold them together. There are three types: tight junctions, desmosomes, and gap junctions. Tight junctions occur when the integral proteins of adjacent cells fuse together. We get an impermeable barrier between the two cells. This keeps molecules from passing through the extracellular space. Basically, this is a waterproof kind of seal, like a Ziploc seal. The epithelial cells of the digestive tract have tight junctions between them. This keeps material in the digestive system, like digestive enzymes, from oozing between the cells and digesting underlying tissues. It also helps keep bacteria that are naturally in the digestive tract from getting into the tissues. Here we see an example of tight junctions. We have these membrane proteins that are fused together, giving us that Ziploc waterproof seal. Desmosomes are anchoring junctions. These are sort of like Velcro spots. We have button-like protein plaques in adjacent cells, and then we have linker proteins that span the gap between the cells and hold these plaques together. This helps distribute tension throughout the cellular sheet and reduces the chance of tearing if the cellular sheet is stretched. We find this in tissues where there's a lot of mechanical stress, things like the dermis of the skin and the heart tissue. So here are the desmosomes, these protein plaques, and the linker proteins. This would allow for a lot of twisting and turning, and this cell junction would not break. We do have some intercellular space, so molecules can get in between the cells. And the final membrane junction are the gap junctions. Here we have transmembrane proteins called connexons. These form channels that will allow small molecules to pass from cell to cell. Ions, simple sugars, and other small molecules can quickly go from one cell to another. Gap junctions will allow for electrical signals to be very rapidly passed from one cell to the next cell. We see this in cells where we have to have rapid communication. Cardiac muscle cells and smooth muscle cells, for example, use gap junctions in order to conduct the electrical signal that makes them contract. And here you see gap junctions, the channels that will allow the molecules to move from cell to cell. When the cells of a tissue need to all work together spontaneously, we will see gap junctions between the cells. Since the plasma membrane is our boundary between the inside and the outside of the cell, we have to be able to transport materials across the cell. Cells are surrounded by interstitial fluid, which contains thousands of substances. It's basically a nutrient soup. There are amino acids, sugars, fatty acids, vitamins, hormones, salts, and waste products out there in the interstitial fluid. Healthy cells need materials from the interstitial fluid and need to put materials into the interstitial fluid to get them out of the cell. The plasma membrane is selectively permeable or differentially permeable. They mean the same thing. Permeable means things can get through. The fact that it's selective means that it allows only certain things in. What the cell needs is allowed in, other things are kept out. There are two general types of transport mechanisms. One group of transport mechanisms are passive. No energy is used. These happen simply because of normal physical procedures. Diffusion, and we have three types of diffusion: simple diffusion, facilitated diffusion, and osmosis, and filtration are passive transport mechanisms. The other general type of transport mechanism is active transport. Here, the cell has to spend some ATP to move molecules across the membrane. We have pumps, and then we have vesicular transport. And vesicular transport can be endocytosis, taking material in, or exocytosis, taking material out of the cell. The process of diffusion is driven by a concentration gradient. Molecules move from an area of high concentration of molecules to an area of low concentration of the same molecule. Diffusion can occur if the membrane is permeable to the molecule in question, and diffusion occurs based on the size and solubility of the molecule in question. Just diffusion in general. If we were to place a dye pellet in this water, we would eventually see it begin to spread through the water. This is because molecules, even in solids, are in constant motion, and as they move, they start to bump around, and they will bump into the water molecules. Smaller molecules will move faster, and if we heat things up, this will occur faster. Eventually, the molecules will be evenly distributed in a substance. When this happens, we say we have reached equilibrium. So how does simple diffusion apply to a cell membrane? Again, the molecules will move along a concentration gradient from higher to lower. In the case of simple diffusion in the cell membrane, nonpolar lipid-soluble molecules can diffuse directly through the lipid bilayer. Nonpolar lipid-soluble molecules would be much like the phospholipid bilayer. We say like dissolves like, lipid dissolves lipid. So if we have a lot of lipid-soluble molecules outside the membrane, they will literally dissolve through the membrane to get into the cytoplasm. This process is nonspecific. It's based strictly on the size and solubility of the molecule. Carbon dioxide and oxygen are both lipid soluble, as are the fat-soluble vitamins A, D, E, and K. Those sound like things we would want to get into or out of the cell readily. But alcohol, which is toxic to the cell, is also lipid soluble. So if the cell is bathed in alcohol, it will take up alcohol, which could be fatal to the cell. So simple diffusion has no selection, just whatever is lipid soluble and small enough gets in. In facilitated diffusion, molecules are still moving along the concentration gradient from higher to lower, but we have to have either a carrier protein or a channel protein. These are both going to be transmembrane proteins, those integral proteins that cross the entire membrane. Because proteins are involved and proteins have specific shapes, we have molecule recognition. So facilitated diffusion will have some specificity. Things like glucose, ions, and amino acids typically travel by facilitated diffusion. Facilitated diffusion is also a little quicker than simple diffusion. These molecules have special doorways to get through, so they don't have to wait to move through the membrane. In the case of a carrier protein, we're going to be transporting specific polar molecules through the membrane that are too large for any of the channels. This is typically how we move glucose, ions, and amino acids. When the molecule binds to its carrier protein, it causes the carrier protein to change shape. That is, it flips from one direction to the other, and it allows the molecule to enter the cell. We're still going from higher to lower. Now, if all of the carrier molecules are busy, even though we have a concentration gradient, more molecules out here than in here, these molecules have to wait their turn. They have to wait until there is a carrier available to take them over. Channel proteins will selectively transport certain ions or water. Here we have specificity that's based on the size of the pore, the little hole in the membrane, or on the charge of this membrane. So if this is positively charged, negative ions would go through. If this were negatively charged, positive ions would go through. Water channels are distributed all over the cell membrane because water can move freely back and forth between the intracellular and extracellular fluid. Water channels are called aquaporins. We do have two types of channel proteins. We have what are called the leakage channels. These channels are always open, molecules are always moving in and out of them. But we also have some gated channels. Gated channels are controlled by chemical or electrical signals. Usually they're closed, but under certain circumstances they open briefly to allow ions or other molecules to enter the cell. Osmosis is a very special type of diffusion. It's the movement of water molecules across the membrane using those aquaporins. Water is always moving back and forth across the membrane, but we will have a net movement of water, that is, more water will move in one direction than the other, if we have unequal concentrations of molecules on either side of the membrane. Now, normally, if there are unequal concentrations, you would think diffusion would take over. But remember, not all molecules can get through the cell membrane. So if the membrane is impermeable to the solute molecules, we move the solvent, we move water. Now here is a case where we have an uneven concentration of molecules on two sides of this membrane. This membrane is permeable to these molecules, and so over the course of time, the molecules redistribute themselves in the two solutions on either side of the membrane. In this instance, however, we still have the same inequality of molecules, but these molecules can't get through the membrane. In other words, the membrane is impermeable to these molecules. Mother Nature doesn't like this inequality, and so water, which can move freely through this membrane, will move over until we have an equal distribution of molecules or particles on both sides of the membrane. In other words, this might be 1 molecule or particle per milliliter, and it's the same on both sides. In osmosis, water moves across the membrane when the solute particles can't diffuse because the membrane is impermeable to them, and it will occur until the number of particles per unit of solution, say per milliliter, is equal on both sides of the membrane. As a general rule, charged particles do not diffuse readily. Fluids that have different particle concentrations, where the particles cannot diffuse across the membrane, are said to have osmotic pressure. That is, they pull water into them. That's what osmotic pressure is. It's that pull of water across the membrane when we have non-diffusible solutes. Osmolality is the measure of osmotic pressure. Interstitial fluids and intracellular fluids have to have the same osmolality. They have to be equal. That's part of what Mother Nature wants. And that osmolality is about 0.9% of a sodium chloride solution. Water will move across a membrane to maintain homeostasis of the interstitial fluid and the intracellular fluid to keep that osmolality the same. Tonicity is simply the ability of a solution to change the shape of a cell by changing its water content. We call the shape of the cell its tone. There are 3 types of tonicities: isotonic, hypotonic, and hypertonic. In an isotonic solution, the concentration of the solute is the same inside and outside of the cell. Since we said the tonicity of intracellular and extracellular fluid was about 0.9% sodium chloride, let's put that in a beaker, and then we will drop a cell in, and the cell has that osmolality of 0.9% sodium chloride. Since there is no difference inside and outside the cell in terms of osmolality, we'll have water moving across the membrane, but it will move equally in both directions. There will be no net movement of water, and the cell will not change its shape or its tone. In a hypotonic solution, the solution is less concentrated than the cytoplasm. So this time, let's put about a 0.5% sodium chloride solution in our beaker. We'll still drop our cell in. The cell still has an osmolality that's equivalent to 0.9% sodium chloride. Now we have an inequality. Neither sodium nor chloride can diffuse, and so we're going to have to alter this inequality using water. Since it's more concentrated inside the cell than outside, water will move into the cell. This will cause the cell to swell, and it may even rupture or lyse. In a hypertonic solution, the solution is more concentrated than the cytoplasm. This time our beaker will contain 2% sodium chloride, and when we drop our cell in, it still has that osmolality of 0.9% sodium chloride. Again, the solute cannot diffuse, so we can't use diffusion to help us with this inequality. We're going to have to use water. This time it is more concentrated outside the cell than inside, so water will leave the cell to try to dilute the external fluid. This will mean the cell will shrivel up. Literally, the cell is dehydrating. Now these are red blood cells. This first one is in an isotonic solution. That's the normal size and shape of a red blood cell, so there's no change. When we put the red blood cell in a hypertonic solution, it's going to lose water. It's going to shrivel up. In the case of a red blood cell, we call this shriveling crenation. And if we put the cell in a hypotonic solution, it's going to take on water. It's going to swell up and potentially could rupture or lyse. There are various homeostatic imbalances that occur with body fluids. One is edema. In edema, fluid collects in the tissues. This can be treated by administering a hypertonic solution. We make the blood more concentrated than normal. This will pull fluid out of the tissues and into the blood to try to dilute that hypertonic solution. Individuals with severe dehydration need fluid. We can administer hypotonic solutions to them to rehydrate the tissues, but this is very tricky and very risky. It's really better to rehydrate them by just having them drink water. Whenever IV fluids are administered, we always use isotonic saline or 9/10% sodium chloride. Sometimes accident victims have an IV started at the site of the accident, not so much because they need fluids, but because they want to keep an open vein. As people go into shock after an accident, their veins constrict, and should they need intravenous medication, it will be very hard to administer it. If we give them a little isotonic saline, it won't hurt them, and it will keep that vein open. The last of our passive processes is filtration. Now this is driven by hydrostatic pressure. We still have molecules moving along a concentration gradient. They're going to move from higher to lower, but we're going to have more pressure on one side of the membrane that's going to force fluid through the membrane. This is a non-selective process. It's only limited by the size of the pores in the membrane. In other words, you can't stuff a basketball through a hole that will only take a baseball, but we can stuff baseballs and ping pong balls and tennis balls through that same hole. We use filtration when we make urine, and also this is how we move materials from the blood to the tissues through the capillary walls. If the plasma membrane is severely damaged, then substances move freely both in and out of the cell. We don't maintain that barrier, and this compromises the various concentration gradients. Burn patients have their cell membranes compromised. They lose precious fluids, they lose proteins and ions that weep from these damaged cells. And if they're not careful, they dehydrate very rapidly and get electrolyte imbalances, which can be fatal.